Chapter 1, Section 12. Koch, Infectious Disease, and Pure Cultures. Robert Koch, late 1800s into the beginning of the 1900s. Late 19th century, beginning of the 20th century. He was a physician and one of our first true microbiologists. Okay? He was the first that really gave us what we now call germ theory. That diseases not caused by, bad air, not caused by, humors in your body being out of whack and things like that. But a disease will be due to a specific microbe. For him it was bacteria, but now we know it's bacteria, fungi, viruses, parasites, and whatnot. He identified the causative agent. Okay, when anyone uses the term causative agent, this is the cause of a specific disease, a cause of a specific agent. We call it the causative, or specific infection. We call it the causative agent. He found the cause of anthrax, Bacillus anthracis, the cause of tuberculosis, what they called back in his time consumption. We now know it as Mycobacterium tuberculosis, and he found what caused cholera. And I'm drawing a blank on the bacteria right now, sorry. Because of him being the main proponent, the guy who really pushed through this idea of germ theory, that germs, bacteria, viruses, fungi, parasites are what cause disease. He came up with this idea of postulates, this checklist that you have to meet before you can definitively say, yes, I have found the cause of a disease. You've got to remember, look, late 1800s, the second half of the 19th century, beginning of the 20th century. Second half of the 19th century, pre-Civil War to post-Civil War. Robert Koch was a German physician, German microbiologist. Okay? In Europe, just like here in the US, it was, for all intents and purposes, wide open when it came to studying diseases. It was wide open. Basically the wild, wild west when it came to medicine. At the time, people, every day, every week were, I found the cause of this disease, I've found the cause of that disease. And then they would try to sell you something that would be used to treat it. Okay, this is the time of the snake oil salesman, which actually happened here, yes, in the West. We see it in the movies. But it also happened in Europe, happened in Asia also. Thing is, what they would do is they would, some physicians, some would-be microbiologists, found a patient, had a patient cough because they had, pneumonia, have them cough in a, in a tube, and then they would go and they would try to isolate some bacteria. And by God, I isolated a bacteria. Here we go. I found the cause of pneumonia. Which is why when you step back and you look, there's quite a few bacteria out there whose genus, different genuses, but all the species are pneumonia, Klebsiella pneumonia, Streptococcus pneumoniae, and so on. They didn't cause pneumonia. They were just isolated from a patient with pneumonia. But even though that would-be microbiologist at the time declared, I've found the cause. Koch addressed this problem. He did so by developing what we now refer to as the Coax Postulates. This, and I'll show it in a slide or two, this checklist. If you can check off all 4 of these postulates, you have found the definitive causative agent for that disease. Finding the causative agent for anthrax, consumption, cholera, 3 of the big killers at the time in Europe, in Asia, in the Americas, that, yeah, your name is going to be remembered by history. Boom. The postulates, yeah, good. Coming with the idea for solid media, what we now think of as agarose. that we use to grow bacteria and fungi on. Yeah, another one of these people. Crazy, stupid, smart. I wish I was half as smart as him. Which is why he won the Nobel Prize. I mean, you add all of this up, yeah. They don't just give that to anybody. Coax Postulates, in a nutshell. Got to meet these 4 criteria. First criteria is that whatever you think the pathogen is, the causative agent, has to be isolated from sick individuals. Everybody who's sick with that disease has to have that microbe in them, and it can't be found in people who aren't sick. Okay? So if it's there, they got to be sick. And if they're sick with it, whatever this disease is, it's got to be in everybody. It can't just be in some of the people have it and some people don't. If they got the disease, it's got to be in them. If they're not sick, it can't be in them. You have to be able to culture it, grow it in the lab, whether in a broth culture, in here as they're showing a petri dish, whatever. You take that microbe and you put it into a healthy individual. Here they're showing a healthy rat. That individual, that rat, has to then get sick with that same disease. Cool. From this diseased person, this diseased animal, you have to be able to isolate the same microbe again. First isolation, second isolation. If you can meet all 4 of these, boom, you win a Cupid doll. You have come up with a definitive causative agent for that disease. Now, we'll talk later in later chapters how nowadays with modern medicine, there are a few problems with this. You know, not every disease, not every causative agent can meet all 4 of these, but we still say this is what causes it. Spoiler alert, think strep throat. Everybody who's watching this, everybody in class has Streptococcus pyogenes in their throat, but not everybody has strep throat right now. Another one is HIV. Yes, we know HIV causes the disease AIDS, but the problem is the HIV that the first individual gets sick with, we can isolate it, we can culture it, and if we are sick enough to give it to another person, that HIV that we take out of them, that virus would not be like the same one, because person to person, within that person, there's going to be mutations. So, this is Hess, a picture of the work by Walter Hess. Hess, contemporary of the time of Koch, was one of these who also became a big proponent of, you know, how developing the augers, developing the petri dishes and things like that, and a means of culturing different things. He took lots of pictures. Those pictures were then being hand tinted. It's basically somebody in with a paintbrush on the negatives or on the picture itself and painted the different things. So he went through and he characterized the growth characteristics. Each of these colonies, this colony, that one, that one, that one, that one, this big one, and these 2 that are overgrown, you see that one there and that one there, all started off with 1 cell. It's the beauty of growing things on agar. Starts off with 1. Can't see 1, but you can see millions of its copies growing right next to it. This I can see with the naked eye. The 1 cell, the 1 spore it started with, no, not so much. When you look at the work by Koch, remember, he came up with the causative agent of TB. How did he do it? Well, he took lung samples from TB patients, pretty sure probably from after they dead, and he noticed, hey, look, here's necrosis. When I stain these samples on slides, here's what I'm seeing. There's, you know, different areas where stuff is accumulating. Well, what is that stuff? Well, it looks like microbes, it looks like bacteria. He cultured it and, hey, look, here's weird stuff. This is not what TB looks like. I can't remember what this is specifically. If this was chunks of material or if this was aggregates of the bacteria. Can't remember what his drawings were. But this is what I saw. I did some of my clinical research on tuberculosis at the University of Louisville. And we would look at, every so often I'd get a sputum sample that I get to look at. Sputum sample. Turns out for TB patients, the best samples to take of their lungs, if they're in an active, active infection right now, is the first cough of the day. When they wake up and they cough, because they've been laying there in a collection of fluid, the collection of mucus in their lungs, they cough it up. That's going to have the highest concentration of bacteria. It's also going to have some chunks of tissue in it because the TB, it's killing off the cells lining the alveoli and that's coming up, so it's going to be a little bloody. You've seen it in movies. You've heard the description of people with TB and they're coughing up blood. Yeah, it's because the bacteria is destroying the alveoli, the epithelial lining of their alveoli. They're coughing that up. They're coughing up those mucus, the bacteria, and chunks of their lung tissue. Cool.